Transcription, the process of copying genetic information from one strand of the DNA into RNA, is termed as transcription. It follows the complementarity and transcription unit. A transcription unit in DNA is defined primarily by the three regions in the DNA. A promoter, the structural gene, and a terminator. Now, DNA dependent RNA polymerase catalyze the polymerization in only one direction, 5' to 3', and 3' to 5', it's template strand, right? Now the other strand, which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil, is displaced during transcription. This strand is coding strand, which do not code for anything, but all the reference points are taken from this strand. Now you see the template strand, coding strand, promoter at the 5' end of coding strand, and 3' end of the coding strand is terminator, and the promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards the 5' end upstream of the structural gene. And transcription unit and the gene. A gene is defined as the functional unit of inheritance. Okay, the DNA sequence coding for tRNA RNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could said to be monocistronic mostly in eukaryotes and polycistronic mostly in bacteria. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or express sequence are defined as exons, right? The exons are said to be those sequences that appear in mature or proceed RNA. Now the exons are interrupted by introns. Now introns or intervening sequences do not appear in the mature or proceed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Okay.